Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 6 for June 2024. Let's begin with the first question. In question 1 they say Saccharomyces cerevisiae is a type of yeast. It is a single-celled organism which is widely used in brewing and baking. The photograph shows yeast cells seen through a microscope. So we can see the yeast cells here. They say most of the ATP produced when yeast respires aerobically comes from oxidative phosphorylation in the mitochondria. Oxidative phosphorylation requires a source of hydrogen ions and the movement of electrons. State the location in the mitochondria where oxidative phosphorylation takes place. It takes place in the cristae, which are the inner foldings of the internal membrane of the mitochondria. And then next they say name one molecule that carries hydrogen for use in oxidative phosphorylation. This is going to be NAD or FAD. They carry the hydrogen and they become reduced. In part B, they say describe an experiment to investigate the effect of temperature on the rate of respiration in yeast using an artificial hydrogen carrier, which is a redox indicator. Examples of a redox indicator that could be used include TTC or DCPIP and others. Since we are describing an experiment to study the effect of temperature on the rate of respiration, the independent variable is going to be temperature, so we have to use at least 5 different temperatures. I gave an example 10, 20, 30, 40, and 50. And then the yeast should be the same species. Of course, this is controlling the biotic factors, so yeast should be the same species. And in this case, I chose using the TTC as the redox indicator. We will incubate the yeast suspension and the redox indicator to the same temperature in different test tubes, and this is going to be in the same water bath to ensure the temperature is uniform before they are mixed. And then to the yeast suspension, we add TTC solution and record the time taken for the color to change from colorless to red. We repeat the experiment at each temperature and calculate the mean time, and then ensure the same concentration and volume of the yeast suspension in all experiments, as well as the same concentration and volume for the TTC solution, which is going to be used in all experiments. And we have to ensure that the pH is kept constant using a buffer. In part C, they say explain why temperature affects the rate of respiration in yeast. Because we are studying respiration, respiration is enzyme controlled, and so as the temperature changes, the rate of respiration is going to be affected. So I said as the temperature increases, there is more kinetic energy possessed by the enzyme and the substrates, so there will be more collisions or faster collisions, and so the rate of reaction will increase. However, at very high temperatures or beyond the optimum, the enzymes will be denatured, and so the rate of respiration is going to slow down. So this brings us to the end of question 1. Let's continue to question 2. In question 2, habituation is an example of learning that can be observed in many different animals. Woodlice are small animals that live in damp places, for example under stones, and in areas with decomposing leaves. The photograph shows one species of woodlouse before and after being gently touched. So this is before it's touched, and that's after it has been touched. We can see it has curled up. Down here they say describe how you could investigate habituation in woodlice. So in this case, they are touching the woodlice, and then the lice are curling into a ball-like structure. So we have to find ways of touching the woodlice. Then we have to ensure that we touch them with the same intensity and maybe at similar time intervals. So the woodlice we experiment with have to be the same age and of course the same species. And then we could record the time taken for the woodlice to uncurl, or we could also count the number of touches required until the woodlice do curl up into the ball-like structure. So I said get woodlice of the same age and mass. Use a suitable, gentle method to touch the woodlice until a response is observed. The touches should be of the same intensity as well as rate. Then we record the number of touches made until the response is observed. You repeat the experiment with other woodlice and record the results. In part B, they say zebrafish are freshwater fish native to South Asia. The photograph shows 2 zebrafish. We can see them here. If zebrafish are exposed to sudden sounds or flashes of light, they show a startle reflex where their body bends quickly and they swim away rapidly. Scientists investigated habituation in zebrafish exposed to a sound. Each fish was exposed to a sound, and the distance traveled by the fish in 0.5 seconds was recorded. Each fish was then exposed to the sound on 4 more occasions. A control group of zebrafish was not exposed to any sound. The table shows the results of this investigation. So we have the group, those exposed to the sound, and those in the control group, meaning not exposed to the sound. And we have the different number of times they were exposed to the sound. So we can see for those exposed to the sound, there is a gradual decrease in the mean distance traveled in comparison to this, because this looks as if it's the same. There is no significant change. Next, they say the mean distance traveled by the fish decreased from 5.2 to 1.5 after 5 stimuli. Calculate the percentage decrease in mean distance traveled. Percentage decrease is equal to the decrease divided by the original times 100, and in this case it's 5.2 minus 1.5 divided by 5.2 times 100, and I got 71.2%. That's the answer to 3 significant figures. And next they say plot a line graph for the results in this investigation. On the vertical scale I came from 0, 1, 2, 3, 4, 5, and horizontally it was 1, 2, 3, 4, 5. If I take you back to the table here, we can see 1, 2, 3, 4, 5, and the vertical scale is of course between 0 to 5.2, or I would say between 1.5 to 5.2, so it falls into the range that I used. On the vertical axis it was mean distance traveled in centimeters, and on the horizontal axis it was the stimulus. So we can see for both results the control group, like I said already, is like there was no significant change, and in the group exposed to sound we can see there is a gradual decrease until the last value, which was about 1.5. Next they say give one way that this investigation should be conducted to avoid harming the fish. Of course, because they were using sound, they should not use very loud sound. And next they say state one abiotic and one biotic variable that could affect this investigation of habituation in zebrafish. The abiotic variable I chose was temperature or the intensity of sound. Then the biotic variable could be age or gender of the fish. The next they say choose one of the variables you have identified, describe how this variable could be controlled and the effect it would have on the results if it's not controlled. The variable I chose was temperature, of course water temperature. And here, how could we control this? It's by using a thermostatically controlled water bath. And the effect it would have on the results if it was not controlled, of course the result would not be valid, because variation in temperature will affect enzyme activity or enzyme controlled reactions. And in this case, meaning if the temperature rose beyond the optimum, then the enzymes are not going to function properly, and so the process is going to be affected. Or if the temperature is too low, then there will be low kinetic energy, and so the rate is also going to be affected. So I said this is going to affect the rate of enzyme activity and hence cause a change in the response. And this could be maybe slowing down the response or increasing the response if the temperature is increased closer to the optimum. So this brings us to the end of question 2. Let's continue to question 3. In question 3, they say the topshell is a marine mollusk widely found in European coasts. It lives on rocky shores and is found in rock pools, on bare rocks, and among seaweed. The photograph shows a group of topshells. We can see them here. Here we're given a scale of 1 centimeter. They say calculate the magnification of this photograph. The magnification is equal to image size divided by the actual size, or what we call the actual object. This is going to be 2.5 centimeters. If you measure that, divide by 1 centimeter, and so I got times 2.5. That was my answer here. In part B, they say a student investigated the types of habitat preferred by these topshells. The diagram shows a tray with 4 different environmental conditions used by this student. We can see there is a rough surface in the light, smooth surface in the light, smooth surface in the dark, and rough surface in the dark. They say the student suggested a null hypothesis that topshells show no significant habitat preference. 5 topshells were placed in each of the 4 areas, and the tray was left for 5 minutes for the animals to move around. The number of topshells in each area was then recorded. This was repeated 3 more times and the results totaled. The table shows the results of this investigation. So here we can see the different areas that were experimented on and then the different trials that were carried out. These results were only recorded for 5 minutes. We can see the total number of observed. Of course, if you add up this, you'll get 12 here, Add up that, you get 15. Add up that, 24, and add up that, we get 29. Here we're given the expected to be 20, 20, 20, 20. So here they say calculate the chi-squared value using the formula. So chi-squared is going to be the total of observed minus expected squared divided by expected. So in this case, from the table, observed minus expected, I'm going to go back to this information and show you how I got it. That minus that, -8, that minus that, -5, that minus that. I think I better put the negatives so that it's easier for you to see them. Let me go back here as well. So I've put the negatives. This minus that, we get -8. That minus that, we get -5. That minus that, we get +4. And this minus that, we get +9. So if we square this, we get 64. Square that, 25. That, 16. And that. And then when we divide that by the expected, it will be 64 divided by 20, we get that. 25 divided by 20, we get that. 16 by 20, we get that. And 81 by 20, we get that. And then I totaled everything to give me 9.3. So therefore, my calculated chi-squared value is going to be 9.30 if I write it to 3 significant figures. And the next part says the table shows some critical values for chi-squared at different degrees of freedom. Now, in this experiment, if you go back, we were given 4 different areas that were experimented on. And so to find the degree of freedom for which we can get the critical value, we have to use the number of categories that were 4 minus 1. So in this case, I'm going to use 3 degree of freedom. And this gives me 7.815 as my critical value. So next they say comment on the results of this investigation. Remember, our calculated value was 9.3. And the critical value from the table is 7.815, so we can reject the null hypothesis. And this shows that these top shelves they have a preference, because remember, the null hypothesis said they have no preference. So this shows that the top shelves have a preference and they prefer darkness to light, because we saw there were more that went to the dark side in comparison to the light. Next, they say justify 3 improvements that could be made to this investigation. If you remember, in the beginning, they used only 5 organisms, and actually they carried out the experiment for 5 minutes per time. So we could increase the time, use more organisms in order to confirm that our results are actually valid. So I said use more than 5 organisms, or you could say more than 5 top shells in each study area. And then you can control conditions like temperature, moisture, and so many more, because these could affect how these organisms move. And then leave the organisms for more than 5 minutes to ensure they have chosen a preferred habitat. So this brings us to the end of question 3. Let's continue to question 4. In question 4, they say when seeds germinate, starch is broken down and used as a respiratory substrate to provide energy for growth. Respiration rate can be measured using a simple respirometer. A student knew that mung beans germinate very quickly and wanted to compare the respiration rate in germinating mung beans with the rates in germinating peas. They formed the following hypothesis. Germinating mung bean seeds will have a faster respiration rate than germinating pea seeds. They say plan an investigation to find evidence to support or reject this hypothesis. The first part of question 4 says describe preliminary practical work that you might undertake to ensure your proposed method will provide quantitative results. If we are planning, we need to find a suitable temperature or optimum conditions, I would say, like temperature as well as moisture for germination to take place. We need to find suitable seeds. How many seeds are we going to use per study, or find the mass of the seeds? We have to find ways of removing any pre-existing microorganisms from the seeds so that any changes in the volume of oxygen are going to be due to the respiration of the seeds only during germination and not the microorganisms that are pre-existing. So I said find suitable seeds to be used and the number of seeds per experiment, find suitable methods to disinfect the seeds, find the optimum experimental conditions like temperature, and find the suitable method to measure the dependent variable, in this case, the volume of oxygen used up. In part B, they say devise a detailed method including how you would control and monitor important variables. Here we need to show what is the dependent variable, how are we going to measure the dependent variable, how are we going to control the variables, how are we going to ensure that the conditions of the organisms used are the same. So I say the dependent variable is the volume of oxygen used per time. You need to set up the experiment using mung bean seeds and pea seeds. Of course, use the same number or mass of seeds. You can sterilize the seeds to remove any pre-existing microorganisms, because pre-existing microorganisms will also carry out respiration, and we want to ensure that the respiration that takes place is only due to the germinating seeds. So we set up the experiment in a respirometer and use soda lime or potassium hydroxide to absorb the carbon dioxide. This is because CO2 is produced during respiration and is a gas, so if it becomes part of the air within the respirometer, then it's going to interfere with our measurement of the volume of oxygen used up. So it has to be removed by using soda lime or potassium hydroxide. Use a colored solution and, using a refilling syringe, return the colored solution to its original position, so marking the volume of air used to refill the respirometer. This is going to be the volume of oxygen that is used up during the respiration process. Then we can calculate the rate by dividing the volume of oxygen used per time and allow the seeds time to acclimatize before the volume of gas is recorded. You need to repeat the experiment with each type of seed and control the temperature using a room heater, control the wavelength of light using a light color filter, and record the results in a suitable table. Next, they say describe how your results should be recorded, presented, and analyzed in order to draw conclusions from your investigation. So you record the results in a data table, including labels and units, and include a column for means, of course, because you're going to carry out repeat experiments. Then you plot the results in a suitable graph with labels and units. In this case, it could be a bar graph, and use a suitable statistical test like the t-test to establish significance of the results. And lastly, they say suggest 3 limitations of your proposed method. In this method, it's going to be harder for us to determine the age of the seeds. It's going to be hard for us to remove all the microorganisms that are pre-existing, and it's going to be harder for us to control the heat within the experiment since respiration produces heat. So I say it's difficult to control temperature during the experiment since seeds release heat during germination or during respiration. The seeds can carry out aerobic and anaerobic respiration, and this could affect the results. It's also difficult to control the age of all the seeds used, and some errors could occur in measuring the volume of oxygen used. This is in terms of reading the scale as refilling occurs. So this brings us to the end of question 4 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.